This video lecture will briefly cover the cellular pathways utilized by your cells in the processing of nutrients for cellular metabolism. In the first video, you learned about the major nutrients that are utilized by the cells: carbohydrates, lipids, proteins. There's also the nucleotides, the minerals, and vitamins. We're going to be looking at the cellular pathways that utilize the carbs, lipids, and proteins in order to generate ATP energy or to store those molecules for later usage to generate energy later. As we do this, we're going to be going through our textbook, and we're going to be analyzing the different aspects of the carbs, lipids, and proteins. We're going to be utilizing the carbohydrate metabolism as our central core. This covers the cellular respiration you learned back in Bio 181, the idea that cells take in glucose and oxygen and generate carbon dioxide and water. This is through the process of aerobic cell respiration. This figure from your textbook gives a quick overview showing you the processes of glycolysis, of the formation of acetyl coenzyme A, the citric acid cycle, also referred to as the Krebs cycle, and the electron transport chain. And in the process, glucose and oxygen come in, carbon dioxide and water go out, and in the process we generate a whole lot of ATP. Now, we're going to be studying that cellular respiration and understanding how all of that works together. Now, in a nutshell, very quickly, you're going to be practice drawing this a whole lot. You're going to be taking a look at how our glucose is broken down to that pyruvic acid, which breaks down to acetyl-CoA, which breaks down to carbon dioxide. And you're going to name the pathways along the way and show where ATP is generated and released along the process. Now, you know that down along through the process we're using those electron carriers, NADH, FADH2. Those are going to travel over to the electron transport chain, where oxygen is going to be transformed into water, and that's going to be where we're going to use those high energy electrons from NADH and FADH2 to generate the vast majority of ATP for our cell. Now, this is the bare bones information. You're going to be putting this together, you're going to be filling this out, fleshing it out, practice drawing this many, many times on scratch paper over and over again. Now, as you do this, one of the things we mentioned before is the fact that your body will at times have excess amount of glucose after a meal, and you don't need it all to break down to make ATP, so you're going to store it. Glycogenesis is the storage of glucose into glycogen. This occurs in the liver. And then later on, when your blood sugar starts to drop hours after a meal, now the reverse, glycogenolysis, glycogenolysis, the cutting of glycogen, is where glycogen is broken down into glucoses, which are freed up to then become blood sugar to fuel the cells. Now, if you are low on carbohydrates, let's say you're on a low-carb diet, and your liver has depleted itself of glycogen, and your body needs more fuel. You're going to switch over to lipid metabolism, and you're going to utilize triglycerides that have been stored in your adipose tissue. Breaking down the triglycerides to generate energy is a process called lipolysis. Lipolysis means to cut lipid. Now, in a nutshell, how that works is that your triglycerides break down into glycerol and fatty acids. The glycerol can be converted into pyruvic acid, which can then just simply be normally catabolized down to generate energy. And the fatty acids can be oxidized into acetyl-CoA, broken into 2 carbon pieces. This process is called beta oxidation, and each of those acetyl-CoAs can again be catabolized, oxidized down to release energy to make ATPs. And that's how we burn fat to make energy. One thing you have to be careful about is in the process of beta oxidation, we generate ketones. Now, ketones are a waste product. Your body will try to get rid of them, throw them out through the kidneys. If you're doing a lot of lipolysis, you're going to generate a lot of ketones. Before they can be cleared out by the kidneys, they're going to make the blood a little acidic. Ketoacidosis. This can have an effect on the central nervous system. It can slow down your thinking process. We don't like having patients in ketoacidosis. It's just not a good thing. So you need to be careful if your patient is doing a lot of fat burning, trying to lose a lot of weight really quickly. They need to be mindful of this possible side effect. Now, in reverse, if you have excess carbohydrates and you're not exercising, your body has stored all of the glycogen that the liver will allow, and you have extra blood sugar. What's your body going to do with it? Well, your body's going to break it down to pyruvic acid and acetyl-CoA. There's no reason to go down to the CO2 if you're not working out right now, needing ATP energy. So that's where your body will then actually switch these arrows and will take the excess pyruvic acid and the excess acetyl-CoA to make glycerol and fatty acid, which will then generate triglycerides. That step is referred to as lipogenesis, which is the formation of triglycerides. And that's how you make fat. And so when you're consuming excess carbohydrates and your body doesn't need it for any specific reason, whether it's a nutrient building block or a nutrient energy source, it'll store it as triglycerides. And so lipogenesis is the reverse, where you're going to wind up making fat. Well, let's say that your patient is starving so far that they're out of carbohydrates and they're out of lipids, their body still needs fuel. Well, that's where you finally go down to proteins. Now, with proteins, you have amino acids. And an amino acid is a carbon that has a hydrogen. It has a carboxyl group. It has an amino group, H2N, and it has some side chain. That side chain is made of carbons and hydrogens, maybe some oxygen, sulfurs. It varies from amino acid to amino acid. Well, this is a single amino acid. If your body is desperate enough for fuel, it's going to wind up taking any of the proteins in your body and start breaking them apart. That amino acid is in the way. We're going to remove that amino acid in a process called deamination. That amino group, which is ammonia, which is very toxic in your blood, will be attached to a carbon dioxide and it'll produce urea. Urea is a waste product, but it's not as toxic as the ammonia. The urea can travel to the kidneys and get thrown out as urine. The remainder of this is simply a carbon skeleton, which can then be converted into pyruvic acid, which can be broken down into make carbon dioxide and ultimately make some ATPs. So that's how you get fuel from proteins. It's not nearly as efficient as carbohydrate or lipid metabolism, but if your body is, oh my God, life or death, desperate, that is a way to get some fuel. Now, that was the deamination part of proteins. And so when we take a look at our deamination, the picture here shows you an amino acid. That amino group gets torn off, combines with gets combined with carbon dioxide to make urea and thrown away. Now, there's another process called transamination. This is how you make amino acids. At the beginning of the last video, I talked about the difference between essential nutrients and non-essential nutrients. Essential nutrients cannot be made in your body, but non-essential can. Of the 20 amino acids, your body can make about 10 of those. And the way that you build them is through the process of transamination. In transamination, you take one amino acid that you have excess of, let's do this, that you have excess of, to make another one that you need. So let's say, for example, I I have an excess red amino acid. Lots of these in the bloodstream. And the cap represents that little amino group. Well, I need a green amino acid. I can make the carbon skeleton. He doesn't have the amino group, but I can make the carbon skeleton. So I can take the amino group off of my red amino acid and just burn him up as carbohydrates within my cellular respiration, attach the amino group to here, and I've just made a green amino acid. And so that's transamination. We transfer the amino group from one amino acid to make another. Now, we are limited in which carbon skeletons that our cells can make. Those are the amino acids we'd consider to be non-essential. We can synthesize those. Some of them we simply cannot make the skeleton, so we have to bring those in the diet. Now, as you take a look at these different elements, one last pathway to have you study is something called gluconeogenesis. Neo means new. So gluconeogenesis is the formation of new glucose molecules. When your body is carb poor, you can switch over to lipid or protein for energy, except in the brain. The brain's only fuel source will be glucose. Brain cells only accept glucose. They won't accept anything from lipid, won't accept anything from protein. And so you need to synthesize a brand new glucose molecule. You do this by breaking down triglycerides or amino acids to pyruvic acid. And then you can, through an energetically expensive process, you can convert that pyruvic acid to glucose. It's very expensive. It actually costs more energetically than what your brain cell is going to get by eventually going all the way through cellular respiration. But the example I give to my class is, think of a mama bird who spends all day flying around eating worms, gets back to the nest, and then feeds the baby birds. The fuel that the baby birds get from that meal that mom gives is not the same calories that mom spent all day flying around collecting those, but mom is willing to sacrifice that so that the babies can live. Your body is willing to sacrifice a lot of work so that your baby brain can grow and stay alive. So that's gluconeogenesis. Now, I went through quickly. You're going to practice this over and over again. In the lab portion for this week, you'll see that there is a group of papers. There's a document to download that will have a group of papers, and you'll cut these up into individual little pieces, and these little papers will list out all the different substrates. And so you'll take each one of these and you'll lay them out and you'll sort them in the order that you need to properly fit out all of the correct pathways. And then the second page will have all of the pathways. And so then you will then sort those out. And you will organize all these together to be able to see how everything fits so that you can properly demonstrate cellular respiration. And so that's what I want you to work on, practicing time and time again, making sure that you understand how that works. This will lead us nicely into our class discussion as we learn about different types of diets. We talk about people who are on a low-fat diet, on a low-carb diet, on the high-protein, low-carb, South Beach, Atkins, keto, any of those. We can then figure out how those work through these different pathways. Good luck.